Hi, welcome to Neuroverse, a podcast hosted by Carolina
and Clara, where we discuss all matters from neuroscience to philosophy and beyond. Hi, welcome back to Neuroverse. Today we're going to be discussing ways to interrogate synaptic function, and we're joined by a special guest, Dr. Rachel Jackson. Rachel is a research associate at the Centre for Developmental Neurobiology at King's College London, where she is studying synaptic structure, function, and plasticity in the Juan Barone lab. Her work focuses on visualizing synaptic proteins and events such as neurotransmitter release in real time, and developing novel genetically encoded reporters to do so, as well as understanding how this changes under pathological conditions such as in neurodegenerative diseases. It's also really exciting to be having a lab mate on the podcast for the first time. Welcome to the podcast, Rachel.
Hi, thanks for having me.
It's wonderful to have you on. So to begin, can you tell us a bit about your scientific journey and how you got to where you are and how you got interested in synapses and synaptic function?
Yeah, sure. So I guess my kind of interest in science really stemmed from secondary school and then all through university, but I was really interested in lots of aspects of science. So I took a really broad natural sciences course where I was able to kind of pick and choose different topics. And then from that's where I really developed my interest in neuroscience. But I guess there was probably some hints that I might be interested in neuroscience already, because my dad was actually also a neuroscientist looking at Parkinson's disease.
Oh, wow.
So, yeah, maybe he could have predicted it. Yeah.
Yeah.
But so I got interested in neuroscience and development as well, embryonic development at university. And then I came to the centre to do my PhD in the lab of Britta Eichholt, where we were looking at ways to kind of control protein function in the developing brain. And then from then, I've kind of just been really interested in the development of neurons and circuits in the brain. I got to know Juan while I was working during my PhD. And after a brief stint over in Berlin, where I did a short 1-year postdoc, I came back to the centre to join Juan's lab. And because I'd seen his work, I thought it was really interesting. And that's really where I kind of focused in on synapses and what they do. And that's, where I am now.
Nice. Really cool. And we have had an episode on synapses and about their evolution a bit and function. But can you tell us a bit about why synapses are amazing? And totally unbiased question. No, but like, what do synapses do and what is their function?
So synapses are the specialized structures that occur between two neurons where neuronal communication actually happens. The information from one neuron gets passed to the next. And if you think about it, that's the key process of the brain, right? Inside our brains all the time, there's processing going on. And without synapses, that just wouldn't be able to happen. Mm-hmm. So they're really important for learning, memory, all the processes that the brains do. And they're really tiny, considering that they have such an important function. So I think they're really fascinating.
Yeah, that's really cool. Can you tell us a bit about, I guess, what is known and what is not known about the function of synapses?
Yeah, so there's quite a lot that's known. They've been studied for a really long time. So we know what they look like. So you have a presynaptic neuron, that's the one that's going to release neurotransmitter. And then you have a postsynaptic neuron, that's the one that's got the receptors that's going to receive the neurotransmitter. We know a lot about the neurotransmitters that are involved. So the main ones in the brain are glutamate in the excitatory neurons and GABA in the inhibitory neurons. We know that the neurotransmitter is packaged in vesicles, these little membranous structures that contain the neurotransmitter. And when the neuron is releasing, one of those vesicles will fuse with the cell membrane, and that releases the neurotransmitter. It has to diffuse across a little gap between the cells called the synaptic cleft, and then it will bind to receptors on the postsynaptic cell. And that whole process is really complicated. It's highly regulated. So one of the key signals that regulates that is calcium influx. And that's something that I'm really interested in studying as well. But then there's a whole host of proteins that are involved in kind of which vesicles are ready to release and how they fuse. So there's a lot that we do know, but there's still a lot that we don't understand.
I think it's really cool how complex things are at that tiny minuscule scale, and also I find it so interesting how these electrical signals through like action potentials and synaptic electrical events convert into these ionic events that lead to physical changes, and I just love that. Yeah, so I was just going to ask, what sort of tools do you use to interrogate this synaptic neurotransmitter release?
So one of the main ones that I use is called functional imaging. And so what we do is we make the neurons express a protein that is normally found in those vesicles, those little membranous bubbles of neurotransmitter. And on that, we put fluorophores that respond to different types of activity in the neuron. So one of them is a red fluorophore. It's going to fluoresce in the red channel, and that is responsive to calcium. So we can see that when the cell is active, when it's firing an action potential, that's when calcium comes into the cell. And then on the same molecule, we also put a green fluorophore. So that responds when the vesicle is actually released, so when the neurotransmitter is being released across that synaptic cleft. And we can monitor both of those processes together. And that's one of the key tools that we've developed in the lab and that we use a lot.
Cool. So I read this paper of yours about correlative live cell and super resolution imaging to link presynaptic molecular organization with function. And I thought it was really cool. And I wanted to ask you, again, within the same theme, about the techniques that you use. Can you tell us a bit more about the super resolution microscopy that you used and the 3D storm as well? And can you tell us what those are and what you discovered with them?
Yeah, of course. So storm, 3D de-storm is the technique that we used in that paper. It's a very cool super resolution technique. So super resolution means that basically we can look at structures that are smaller than you can look at with a traditional light microscope. And so the reason that's important is because, as we've mentioned, synapses are really tiny. So a synapse is maybe around 1 micron, but the active zone of a synapse could be only a few hundred nanometers. And that's the area where the neurotransmitter release actually happens. And so when we were doing that paper, what we wanted to look at was how are the molecules of the active zone arranged in synapses, and can we link any of those kind of arrangements that we see to the function of the synapse? And yeah, STORM's a really cool technique where you label the proteins that you're interested in with antibodies, and then you use the fact that you can kind of turn on and off the antibody label that's fluorescent randomly, and then you can kind of build up a picture of where each little dot of fluorescence is, because it's going on and off randomly, different to all of its neighbors. It's kind of like building up a picture with points, like pointillism. So that's what we did, and we imaged some synapses live with that reporter that I was talking about before, and then we fixed those cells, we stained them for the proteins we were interested in, and we used STORM to look at their kind of arrangement, how many there were, how clustered they were. Those are the kind of questions that we can ask with those super resolution techniques.
So what kind of structures can you see at that resolution? You mentioned molecules. Can you see like proteins? Or... Yeah,
so that's the point. So to try and look at how the proteins are arranged. So one of the things that's come out quite recently, not from our lab, but from other labs, is that in this active zone where the neurotransmitter release happens, you get sort of sub areas that are denser. They're like enriched for certain proteins that are important for neurotransmitter release, the ones that kind of control whether or not the vesicle can fuse with the membrane. And it's been found that those areas that are the most dense on the presynaptic side, they have a corresponding kind of enriched density of postsynaptic proteins on the other side. And it means that even kind of at the sub-micron scale, like within the synapse, there's then areas that are really specialized for release and reception of that signal. And if you even kind of misalign those bits, those two parts, then it makes the signal much less effective and efficient at transferring information. And that's called a nanocolumn. And so what we were kind of looking at was, whether we could see clusters of other proteins that are related to release or related to kind of the structure of that area, and see if we could also see kind of clusters.
Mm-hmm. That's really cool. I feel like that really relates to a topic that we talk a lot about in Neuroverse, like the structure-function relationships. So from what I understand, these proteins need to be arranged in a specific way, and then on the other side as well, in order to actually have the release happen and for it to be successful.
Yeah, basically, for it to be at its most efficient, you want everything to be aligned properly across the synapse, basically.
Yeah, really interesting.
So in terms of the nanocolumns needing to be structurally organized, have you found that this is different in certain conditions, like when you have pathological neurons?
Oh, that's a really good question. I haven't actually looked, so I'm not sure if there's any known examples of nanocolumns being misaligned in pathological systems, but in the original paper where they described it, they were able to kind of make misalignments by sort of altering the activity of the neurons on a short-term kind of acute basis.
Wow, they were able to induce structural changes by altering activity of the neurons.
Yeah.
It's really interesting that you're able to, with this tool, image both the presynaptic calcium influx and the exocytosis of the neurotransmitter. Could you talk a bit about how calcium dependence influences exo and endocytosis?
Yeah, sure. So it's been known for a really long time that calcium's crucial for the exocytosis process, and that also it's a non-linear relationship. So you need multiple calcium ions to come in order to cause the release of a single vesicle. And in terms of endocytosis, that is also a calcium-dependent process, but that is a much more kind of recently discovered phenomenon. But calcium is generally important for the whole vesicle cycle, and it's really tightly regulated where the calcium comes in. So you have these voltage-gated channels that, again, we think might be organized on the nanoscale to be in the really close position to where the vesicles need to release. So we think that calcium is very important for that process, and where it is very important.
And so is the main advantage of this tool that you've created that you are able to look at the calcium dependence specifically and not just exocytosis on its own or calcium release on its own?
Yeah, I think that is definitely one of the key new kind of features of that tool, because for example, I've been looking at a mouse mutant that has a deficit in neurotransmitter release. It's a particular protein that has been known to respond to the calcium signal and help the vesicle fuse with the membrane. But by looking at both processes simultaneously, I've actually seen that there does seem to be changes in the calcium influx even ahead of the neurotransmitter release. So by monitoring both those things, I think sometimes you pick up changes that you might not pick up if you were only looking at one or the other.
And what other sort of techniques can you use to look at synaptic proteins?
So we've talked a bit about STORM, and I think that's a really cool technique that you can use a lot. There are variations on other super resolution techniques as well, and different ways of labeling. There's something called DNA paint, where you can look at multiple targets of interest at the super resolution scale. But one of the other ones that I use quite a lot is a biochemical technique. So you can actually make these things called synaptosomes, which is where you use a specific buffer. It has lots of sucrose in as well as some other ingredients. And you essentially break the cells apart, but in a very specific way, so that the pre and the post-synaptic areas stay together and form kind of, they reseal basically where the membrane breaks off from the rest of the cell. They reseal themselves into kind of, again, like two bubbles that are attached, and they're actually still functional. So you can use them for neurotransmitter release studies, or even, you could look at calcium. But the thing that I do with them is that I tend to lyse them afterwards. So that's, again, splitting the cells apart. So I get them and I purify them, and then I split that synaptosome kind of fraction, we call it. We split that apart, and then we can look at what proteins are there. And the reason that's useful is because a lot of the proteins that are at the synapse are quite specific, and if you look at them in the context of all the other proteins in the cell, you might not pick them up because they might be at quite a low level. But when you've enriched your sample for just the synaptosome, you can pick up even the proteins that are not very highly abundant.
Once you've isolated the synaptosomes, what can you do to look at the proteins?
So one of the ways we can look at the proteins just to see what's there is we run them on a gel, and then we do this process called Western blotting, and that allows us to identify which proteins are present and whether or not the levels change between different conditions. But another cool technique that we've been doing a lot is we take a protein that we know is in the synaptosome, and we use an antibody to that protein to kind of capture it, and we then look at what other proteins have been captured at the same time, so the proteins that are interacting with the one that we're interested in. And that's something that we're doing quite a lot at the moment, and then we'll run the same kind of gels to see which proteins were present when we captured our protein of interest.
Cool, it's like taking a snapshot of a synapse.
Yes.
Can you also in that way maintain the proteins between the synapses, like the transsynaptic molecules?
Yeah, I think in theory that's how they sort of stick together, because there are molecules that bind across that cleft that are keeping them together, although it's not really clear how they stay, when the rest of the cell is being broken up, but I assume it is because those proteins are still intact and still binding.
Cool. I remember when I learned about synapses, I always imagined it as like, a gap and like two compartments, presynaptic, postsynaptic. And then one day they taught us about transsynaptic molecules and like how there's actually no space in the brain. It's all filled because it's all filled with like molecules or proteins.
Yeah, it's true. There is a whole sort of forest of proteins sticking out from both the pre and the postsynaptic side that interact. Wow. But there must be a little bit of a gap because the neurotransmitter can get through and can diffuse across the cleft.
So can you talk a bit about what you're using these, like what sort of questions you've answered or are in the process of answering with these amazing tools that you've just described?
Yeah, so one of the main questions was first just to look at how do healthy synapses look in various models, so in rodent primary neuronal cultures, and more recently I've moved to human stem cell derived neurons. And I've also been interested in looking in situations where neurotransmitter release might be compromised. So one of them was this mouse mutant that I was talking about, which is lacking a protein involved in neurotransmitter release. But more recently, and probably most excitingly, what I'm doing is working with these human stem cell derived neurons that are modeling the disease ALS. So ALS is a type of motor neuron disease, but it's also shares a lot of its pathology and some of its genetic causes with frontotemporal dementia, which is obviously something that affects the cortex rather than the motor neurons. So what we're doing is making cultures of these neurons that are like the excitatory neurons of the cortex, or as close as we can make them from stem cells. And then we're using the tools, mostly we're using the functional imaging tools, looking at calcium influx and neurotransmitter release. And we're trying to see if we can pick up early kind of changes in the synapses in those disease model neurons. And if we can pick them up, is there a way that we can rescue them by targeting the proteins that we know are affected in that disease?
That's really cool. Do you know if it's like just a matter of missing a protein, or is it that the proteins aren't arranged in the right way, or is it that the important protein itself is like malformed in some way?
So in ALS FTD, one of the big changes that happens is this protein called TDP43, which is normally a nuclear protein that kind of modifies how other proteins are made. And so when that protein function is lost, you end up with a whole bunch of other changes in the proteins that are the targets of that. And recently it's been found that one of the key targets of this TDP43 protein is called UNC13A, and it's a protein that is involved in preparing the vesicles for the release. So it helps them to dock to the membrane and get in the right place and in the right conformation in order to be able to fuse when that calcium signal comes in. And we know that the problems that happen in ALS FTD are that usually you don't get this UNC13A protein. So it's actually just missing from the neurons that are affected. And that's one of the things that we're trying to now model and see how it affects the synapses and their function.
What would be like the next step? Would it be at all, like for a clinical solution, would it be at all possible to introduce this UNC13A?
So we are interested in trying some of these potential therapeutics, and the approach that we're taking is actually based on the fact that the issue with the UNC13A protein happens at the previous stage. So the protein gets made from something called RNA, and normally that RNA needs to be processed. So when it's first made, it's not the complete form that it should be. There are some bits in there that shouldn't be in there, and there are some bits that need to be kind of rearranged before the RNA is in the right form for it to produce the functional protein. One of the issues that happens in ALS FTD is that the RNA is not processed properly, and you actually get a little bit extra being made that shouldn't be. So when you get this little extra bit in the RNA, it basically means the RNA gets degraded because the cell kind of recognizes that it's wrong, and therefore you don't have the RNA, and you don't then have the protein being made.
Okay.
So the technique that we need to use is to stop this little bit of extra RNA being included. And there are various ways you can do that, which are called antisense oligonucleotides. And basically they bind to the region where this mistake happens. And so the idea is that if you can stop that mistake from happening, you'll get proper RNA, and then you'll get proper protein.
Nice. That's really cool.
Would there be another way to alter neurotransmitter release that isn't starting from the bottom up, like that isn't starting from fixing the protein? Could you do some sort of compensatory mechanism artificially to like promote the neurotransmitter release?
Potentially, yes, but I guess you'd still have to try and express some other protein that could maybe take over the function of UNC13A. So there is an UNC13B, and we know in mice that some synapses use UNC13B rather than UNC13A, but it's not so clearly understood in humans what the two forms are doing. And you'd still have that problem that you've got to then deliver that protein to the cells that need it and make sure it expresses it in the right kind of amounts.
And also, I'm just thinking about scales here and how it's all happening at such a small scale. And do you think that these deficits in neurotransmitter release in ALS or FTD and all the potential other neurological disorders, that this is kind of a causal mechanism for many of the other phenotypes that we see in these disorders?
You mean the synaptic function changes are a causal phenotype?
These changes at the synaptic level lead to other phenotypes that we see at the cellular level and at the network-wide level, and how circuits act and behave. And
yeah, I think it's possible. So there is a school of thought that if the neurons aren't forming the right kind of functional connections, and then they don't have the right levels of activity, then that can kind of cause problems with the neuronal health, and that could lead to cell death. I think in some disorders and diseases there's probably other mechanisms that are also causing the cell death, and with ALS FTD we know that there are a lot of other targets of TDP43 that are also going to be changed and have implications for the cell health and for the cell function. So I think the synaptic phenotype is important because it could be an early sort of it could lead to early symptoms, for example, and therefore if we can treat those, if we can kind of bring back synaptic function, that might help patients, for example, with their, in the early stages of the disease. But whether or not it would actually stop the degeneration of the neurons is not yet clear, because we don't know if it's causal, and there are probably other mechanisms.
And do you know in general if a lot of diseases that involve synaptic deficits, if this largely influences the release of neurotransmitters, or does it mainly influence, some other synaptic mechanisms? Like, I guess there's also postsynaptic mechanisms in receiving the neurotransmitter, or like the endocytosis of the vesicles, or...
Yeah, as you say, there's definitely various places and various aspects of synaptic function that can be affected. So neurotransmitter release is definitely one of them, and there is sort of a growing understanding that maybe there is a sort of a group of disorders where there are similar phenotypes and similar genes that are all involved in the synaptic vesicle release, that are starting to be kind of grouped together by clinicians. But there's also changes that can happen in synaptic number. There's changes, as you say, on the postsynaptic side, and whether or not the cell can actually receive the signal. So even if a cell is giving out healthy neurotransmitter release, there might be problems on the postsynaptic side. And then there's also problems potentially with the cell turning those signals into then an electrical signal, which is how it's then transferred from, within that postsynaptic cell to the next synapse. So there's definitely a whole host of changes that could happen.
Yeah. And on that subtopic of like heterogeneity, I also realized that we're kind of generalizing synapses, but of course there's also many different types of synapses, and like largely they are mainly divided into like inhibitory versus excitatory. So is all of the stuff that you've been talking about, and is your research mainly focused on the excitatory synapses?
Yes, pretty much almost exclusively I've looked at excitatory synapses, but inhibitory synapses are also very interesting and play a lot of roles in neurological diseases.
Do you know if they act very differently at that kind of fundamental neurotransmitter release level?
Yes, so the process of release is basically identical in the two types of neurons. The difference is really in the proteins that load the vesicles with neurotransmitter, because you've got different neurotransmitters, and then obviously in the receiving of the signal you have different receptors because it's a different neurotransmitter.
Yeah, and I imagine that, going back to the nanocolumns, I imagine that the misalignment and potentially like the misfiring or the mistransmission of the neurotransmitters also affects the excitation and inhibition balance, like across the brain, which also affects a lot of other neurological disorders such as epilepsy. And I was wondering if you know or think that there are any, let's say, machine learning based tools that can help in this kind of research in terms of understanding the protein arrangement or structure. So we've all heard about AlphaFold, but I feel like maybe that's not applicable for this kind of research, just because it's more about predicting the structure of a specific protein, when I think in your kind of research it's more about the protein to protein interactions and the arrangement between proteins.
Yeah, definitely. And I think it's an area where machine learning could really help, because it is a really highly complex environment. There's hundreds of proteins, there's different ways they can be arranged, but I don't know if we have enough data at the moment to even be able to train a machine learning algorithm. I think we're still in the stages of trying to understand ourselves how the proteins are arranged and how many there are of each type, and how heterogeneous the different populations of synapses are. So obviously there's differences in the excitatory and inhibitory synapses, and they'll probably have different proteins as well. But even within excitatory synapses, we know that synapses can come in a huge range of shapes and sizes. You have strong ones, you have weak ones, so ones that are very good at releasing neurotransmitter release, and then you have ones that don't release very often and only do so occasionally. So how the arrangements kind of relate to that, we don't have enough data, I don't think, at the moment. But once we start getting the data, maybe then that's when we need to get those machine learning tools.
Cool, yeah.
I think that also highlights the advantages of your kind of approach, because it's looking really at the dynamics, not just at the levels of protein or just the location of the proteins, but it's actually imaging them in real time with some live imaging.
Yeah.
So would you agree that maybe previous research was more focused on understanding like what proteins there are at synapses and kind of taking that static approach to looking at the proteins? And would you say that the future of synaptic research is more trying to understand the dynamics of the system?
I think definitely the dynamics of the system is kind of an important avenue for research, because we know that synapses are also plastic, so they change their strengths and their structures based on prior activity that the neuron has experienced, and an ongoing activity. So I think understanding the synapse as a dynamic structure is really crucial.
So in neuroscience, we're often limited by either like a high temporal resolution or a high spatial resolution. So we often either have really good imaging tools that let us know exactly where the cells are, or we have other tools that tell us exactly what the firing frequency is. Do you think in biochemistry or when researching in synapses, there's a similar kind of duality where there's like a really significant limiting step?
Definitely different techniques have their own limitations. So the imaging technique that I use, I've talked mostly about presynaptic imaging, but that can give you, for example, the location of the synapse along the axon, how far it is from the soma, what its neighbors are doing, but it is slightly limited temporally. So there is a limitation in how quickly the fluorophores can react and how quickly I can image when I'm trying to look at two channels at once, and depending on how dense the cells are, how many synapses I can look at once. A complementary technique that one of my colleagues uses a lot is electrophysiology. So they would look at postsynaptic events onto a cell and they'd be able to see how many times that cell had received a synaptic input, and that has a much higher temporal resolution, but they don't have the spatial resolution because they don't know where those synapses are. And so I'm now trying to complement that technique, again going back to imaging and looking at the postsynaptic side now. So I express my reporter, and in this case I'm using a reporter called iglutinib 3, and what that does is fluoresces in green whenever there's glutamate, and glutamate is that excitatory neurotransmitter. And so I express it in the postsynaptic cell, and then I can look along the dendrites of that cell, and every time I see a flash I know that that cell has received a synaptic input. So it gives me that spatial resolution that I get from imaging. Again, it's not quite as fast as recording a cell electrically, but these probes are getting faster and faster, so we're really getting to the point now where we think we can see the individual events happening in real time.
Cool. Can you combine that with electrophysiology and look at the electrical activity at the same time as the glutamate?
Yes, we haven't done it yet, but we are planning to. That would be a very cool experiment.
Yeah.
Nice.
So to end the episode, in collaboration with the Center for Developmental Neurobiology and the Center for Neurodevelopmental Disorders, we have collected some questions from our Dev Neuro Academy students, who are a group of year 13 students who are interested in getting some lab experience and spend some time here at the center. So these are questions they've written down that they have for neuroscientists, so we thought we would ask you one today.
Okay.
So we have one from Kate, and the question is, how have you dealt with failure in research?
Wow. I mean, it's a great question because failure is a huge part of research. Mm-hmm. There are, a myriad of ways that your experiment can fail. And that can range from something like a technical issue that you have to sort of figure out and troubleshoot, whether it's a problem with your microscope or a problem with the tool that you're using. And then there's sort of the failure at the next level, which is that you might have a hypothesis and then you test it out and actually nothing has changed or nothing's happened or it doesn't happen the way you think. And I think that being able to deal with that and take those failures as sort of a learning experience is really important as a scientist. So for example, just being able to sort of learn to troubleshoot is a really key aspect of being a scientist. Being persistent and being able to kind of change your perspective based on whatever the results might be, whether they're a failed experiment or a successful experiment, you have to be open to that. And I think that that's something that you do learn just through the process of going through it. And the more science you do, the more failures you're going to have, but it's completely normal. And I think, again, just having a really great team of co-workers and people that you can discuss the problems that you're having with, people that you can discuss the problems with your research with, then that's really helpful. And I think that's how you can get through those failures.
I think that's a great answer.
Yeah.
And
very
also
well said.
reminded me why I'm doing PhD,
Yeah.
to gain all of those qualities you just mentioned.
Yeah, resilience, persistence,
Learning
all
how
of
to
them.
troubleshoot.
Yeah.
Yeah. Okay, thank you so much for joining us today and talking about your research and your expertise, Rachel. It's been really nice having you.
Thank you for having me. It's been really fun.
Yeah, it's been an absolute pleasure. Yeah, it's so fascinating understanding how all of these different proteins work together in the synapse.
Yeah, and how complex the dynamics are at the synaptic level. And I hope you enjoyed listening to this episode. If you enjoyed the episode, please share it with your friends and family and follow us on social media at neuroverse_pod on Twitter and Instagram, and check out our website neuroversepod.com for links to all of the resources for all of our episodes.
We'd like to thank the Biochemical Society for supporting Neuroverse, a podcast about science and philosophy. This episode is part of our series to disseminate research done in biochemistry.